Hello, good morning all. This is Dr. Shaima, and in today's class, we'll be dealing with the endocrine hormone glucagon. Hope you'll have a notebook in hand so that you all can take down the lecture notes. So we begin with the class. So now glucagon, if you see, glucagon was discovered very shortly after insulin. But its physiological importance, it took a lot of time, for the scientists to demonstrate the physiological importance of glucagon. But it's very clear that glucagon is an important regulator of intrahepatic glucose and free fatty acid metabolism. Glucagon has exactly opposite metabolic action to that of insulin. So, if you all have been thorough with our previous class on insulin, understanding glucagon is absolutely not an issue. So before you all actually start studying or listening to the class on glucagon, please make sure that insulin is in and out thorough for you all, because this hardly takes 15 minutes to understand and study glucagon then. So what I was trying to tell you all was insulin regulates the intrahepatic glucose and also the metabolism of free fatty acids. And its actions or the metabolic actions on the cells of glucagon are exactly opposite to that of insulin. So you can call glucagon as a pro-diabetogenic and a ketogenic hormone. So glucagon is a pro-diabetogenic and a ketogenic hormone. So let's go to the second slide. The second slide gives us a brief intro into the structure and structure of glucagon, and also exactly where the hormone is secreted. So when you look at the structure of glucagon, it's basically a single straight chain peptide hormone. It's just a single chain with around 29 amino acids. So glucagon is a single chain peptide hormone and it contains 29 amino acids. The glucagon gene is located on human chromosome number 2, and it's this human chromosome number 2 which directs the synthesis of a pre-pro-glucagon in the alpha cells of the pancreatic islets. And you know that the pancreatic islets, the alpha cells which secrete glucagon, constitutes around 20 to 25% of the islet cell mass. And under the direction of the human chromosome number 2, the alpha cells secretes pre-pro-glucagon. Glucagon is also secreted in the intestine, which acts as a local hormone, and it's found in some parts of the brain. And in the brain, this glucagon, which is secreted by the intestine, we call it as the enteroglucagon. It acts as a neurotransmitter in some parts of the brain. It's a very local neurotransmitter. So glucagon secretion, like insulin, happens by exocytosis, and the secretion is inhibited if the alpha cell calcium concentration goes down. I think those 2 points are very similar to that of insulin, isn't it? The glucagon secretion happens by exocytosis, and the glucagon secretion by the alpha cells can drastically go down if the intracellular calcium concentration of alpha cells goes down. So basically glucagon secretion is a calcium induced exocytosis. Now if we look at the mechanism of action, glucagon acts on the surface receptor of the target cell, but through a cyclic AMP second messenger system. So a second messenger system is involved here, which is cyclic AMP here, and glucagon exerts its actions on the tissues by binding on the surface receptors, and the mechanism action is modulated through cyclic AMP second messenger system. Now let's go to slide number 3. where it mentions the actions of glucagon. Before we really move on to the effect on the carbohydrate metabolism, protein, and then fat, we need to have some idea on how exactly glucagon acts on our body metabolism. Glucagon has anti-insulin like actions in all aspects. Whatever insulin was doing, glucagon does exactly the opposite. So what glucagon does is, if you sum up its effects on carbohydrates, proteins, and fat metabolism, you can see that it facilitates the mobilization of nutrients by decreasing their storage. Okay? So it especially facilitates hepatic glucose output and also ketogenesis. Please keep this in mind. Glucagon has an anti-insulin like actions in all aspects. It facilitates mobilization of nutrients by decreasing their storage. And it specially facilitates hepatic glucose output and also ketogenesis. So let's first see the effect of glucagon on carbohydrate metabolism. Glucagon mainly acts on the liver and the adipose tissue. It increases the blood glucose by the following 2-3 mechanisms. We'll go in detail. But please keep in mind that glucagon, all the actions of glucagon, whatever we are going to discuss in the coming up slides, you need to keep in mind that the glucagon's action is getting more focused on the liver and the adipose tissue. So you know that glucagon increases the blood glucose. Let's see by which all mechanism. Glucagon stimulates, it's put as point number 2 there on the PPT. It stimulates glycogenolysis in the liver. Now how does it stimulate glycogenolysis? By activating the enzyme glycogen phosphorylase. Now please keep in mind that it does not cause glycogenolysis in the muscle. So glycogenolysis is caused by glucagon in the liver by activating the enzyme glycogen phosphorylase. Now point number 2, it promotes gluconeogenesis. Now how does it promote gluconeogenesis? By activating a gluconeogenic enzyme, especially the enzyme phosphoenol pyruvate carboxykinase. Please remember the name of the enzyme. Gluconeogenesis is promoted by glucagon by activating the enzyme phosphoenol pyruvate carboxykinase. It promotes hepatic proteolysis and it supplies more amino acids for gluconeogenesis. That's how the mechanism works. So please remember that normally the metabolic actions of glucagon on muscle and adipose tissue are not very significant. Okay. Now let's go to point number 2, where the slide tells us it's the effect on protein metabolism. So if you look at the effect on protein metabolism, it increases amino acid uptake by the liver and hence it promotes gluconeogenesis. Hence glucagon lowers the plasma amino acid concentration. So please approach that point as, how does glucagon lower the plasma amino acid concentration? How does it lower? It increases the amino acid uptake by the liver and it will promote gluconeogenesis in the liver. The result would be a lowered plasma amino acid concentration. Now let's go to slide number 4, where point number 3 tells us about the effect on the lipid metabolism. So to brief up about glucagon, glucagon is a powerful lipolytic agent. It also promotes ketogenesis in the liver. In the liver, excess free fatty acids are oxidized, which results in energy production and ketone body production. That's how glucagon increases the energy production and the ketone body production by how does it do? The free fatty acids, the excess of the free fatty acids in your bloodstream are getting oxidized and increased energy production and ketone body production happens in the body. Glucagon prevents the synthesis of triglycerides. And what happens is the spared free fatty acids are used for the formation of ketone bodies. Glucagon increases calorigenesis. Calorigenic action of glucagon is not due to increased blood glucose, but it's due to the hepatic deamination of amino acids. This point you need to keep in mind because this can be asked for your theory viva. So glucagon is a calorigenic hormone, or it increases calorigenesis. And the calorigenic action of glucagon, it's not due to the increased blood glucose levels, but it's due to hepatic deamination of amino acids. Due to glucagon's ketogenic and hyperglycemic actions, we call glucagon as a hormone of energy release. Now we go to the next slide, the slide number 5. Now a point which you need to keep in mind here is glucagon, is calorigenic, but glucagon can act as a calorigenic hormone only in the presence of glucocorticoids and T4, which is released by the thyroid gland. Okay, now glucagon. Now let's go to point number 6. Glucagon in supraphysiological concentration can increase the myocardial contractility. How does it do? Glucagon in supraphysiological concentration, when I tell you it increases the myocardial contractility by using the cyclic AMP levels or by increasing the cyclic AMP levels in the myocardial cells. So glucagon, when you administer glucagon in supraphysiological concentration, that will increase the cyclic AMP levels in the myocardial cells, which results in an increase in myocardial contractility. Now point number 7, it stimulates the secretion of growth hormone, insulin and pancreatic somatostatin. Point number 8, glucagon may be secreted in the central nervous system. When it is secreted in the central nervous system, it acts locally in the brain by regulating a person's appetite. Now, there's a little more input I need to give you all on the insulin-glucagon ratio. These points are not mentioned on the slide, so I would want you all to note it down somewhere. Insulin-glucagon ratio can be asked as a 3-marker. Now, if you try to brief up what we have learned yesterday about insulin, you understand that insulin is glucopenic, glycogenic, anti-gluconeogenic, anti-lipolytic, anti-ketotic. I repeat it, insulin is glucopenic, glycogenic, anti-gluconeogenic, anti-lipolytic, anti-ketotic. 5 points I have briefed about insulin. So basically it can be categorized as an anti-diabetogenic and anti-ketogenic hormone because of these 5 points I have told you. So insulin is basically the hormone of energy storage, isn't it? Basically insulin, as it is an anti-diabetogenic, anti-ketogenic hormone, we can categorize it as a hormone of energy storage. But when you look at glucagon, glucagon is a pro-diabetogenic and a pro-ketogenic hormone. So when something is pro-diabetogenic and pro-ketogenic, then glucagon can be categorized as a hormone of energy release. So insulin is the hormone of energy storage, glucagon is the hormone of energy release. Because of their opposite effects, a balance should be maintained between the secretion of insulin and glucagon for maintaining the normal metabolism of or the normal metabolic functions of the body. So this is what you need to keep in mind. Because the actions of insulin and glucagon is opposite, and insulin is a hormone of energy storage, glucagon is a hormone of energy released. Because of their opposite effects, a balance should be maintained in the levels of insulin and glucagon. Only then the metabolic functions of the body can go smooth. So insulin-glucagon molar ratio, we put it as a capital letter I / and a capital letter G ratio in the plasma, is more important than their individual concentration. Normally the insulin-glucagon ratio following a very balanced diet is 3. After an overnight fast, it drops to 1. Sometimes after a very prolonged fast, you can see that the insulin-glucagon ratio drops down to around 0.4. But after a glucose infusion, the value can go up to 30. Now a little peep into the applied physiology. Glucagon excess is produced by, or when there is a tumor of the alpha cells of pancreas, which we call as glucagonoma. Glucaganoma, glucagonoma. But glucagon deficiency appears very rarely in a person's body. That's just for an understanding for you all. So there we wind up with today's class with glucagon. I again remind you all to make your lecture notes which would support the PPTs which I am uploading, and once the study is done, please get your mind maps ready. Thanks for listening, guys. Bye.